Welcome to this week's Deep Dive. We're looking at the most exciting longevity papers published between February 6th and February 13th, 2025.
Yeah.
We're going to be focusing on research with the potential to really shake things up, for people in the longevity biotech world.
Absolutely.
We're really looking for those paradigm-shifting discoveries from trustworthy institutions.
Right, those ones that make you go,
Yeah, the ones you really want to think about for a while.
Yeah, and the ones that are probably going to be impactful for longevity, too.
Exactly. So as researchers and experts, these are the things you need to know about.
For sure. It's been a fascinating week for new research. A lot of really interesting stuff coming out.
Yeah.
There are a few papers that really stood out to me as potential game changers.
Oh, awesome. Well, let's hear about them. Let's start with this one that caught my eye. It's called Longevity Humans Have Youthful Erythrocyte Function and Metabolic Signatures.
Okay.
It was published in Aging Cell on February 10th by Fang Yu and colleagues from the Department of Neurology at Central South University in China.
Interesting.
And this one is really interesting because it kind of challenges the assumption that age-related decline in blood function is just inevitable.
Right.
And they found that individuals with exceptional longevity have red blood cells that function much like those of younger individuals, particularly in their efficiency of oxygen delivery and their ability to combat oxidative stress.
So it's not just that their blood is younger.
Right.
The researchers dug deeper and found unique metabolic signatures in these long-lived individuals' red blood cells.
Yeah, that's what I thought was really fascinating about it.
What are the implications for potential interventions?
I know, right? That's what I was wondering.
Yeah, that's the million-dollar question.
I know.
If we can pinpoint the specific metabolic pathways that contribute to this youthful function, maybe we can target them therapeutically.
Oh, okay.
Imagine being able to enhance the function of our red blood cells to mimic those of younger individuals.
Oh, wow.
It could have a really significant impact on overall health and lifespan.
Yeah.
So this research definitely opens up a whole new avenue for exploration.
Oh, for sure. Okay, so from the intricacies of our blood,
Right.
let's shift to the bustling ecosystem within our gut.
Okay, the microbiome.
Yes, the microbiome. There's this bioRxiv paper from February 6th titled High-Resolution Temporal Profiling of the Gut Microbiome Through the Whole Life of Mice, and this one really dives deep.
Oh, wow.
The researchers, Takayasu and his team from Riken, tracked changes in the gut microbiome of mice from birth to natural death.
Okay.
Over 1,000 days of data.
That's impressive.
I know. That's a lot of data.
1,000 days.
They found that not only does the composition of the microbiome change over time,
Right.
but there are distinct patterns of synability.
Okay.
Some bacteria, the life course species, persist throughout life, while others are more transient.
Interesting.
I found it really striking that there was this correlation they observed between lifespan and microbiome diversity.
Yeah.
So in middle age, greater diversity was linked to longer lifespans. But in old age, it was the opposite. More diversity was associated with shorter lifespans.
Interesting.
It's a bit of a head-scratcher, isn't it?
That's a very good point. It really does challenge that idea that promoting gut diversity is always beneficial for longevity.
Right.
Seems like the relationship is far more complex and dynamic than we thought.
Yeah.
It changes throughout the lifespan. And, it raises some important questions for researchers. What's driving this shift in the diversity lifespan relationship as we age?
Right.
Could it be that a diverse microbiome becomes less important or even detrimental in later life? And how does this knowledge impact our approach to gut health interventions for older adults?
I don't know, but I want to know.
Yeah, me too.
Okay, so from the gut, let's move on to a pair of papers that explore the fascinating realm of epigenetics,
Okay.
the modifications to our DNA that influence gene expression.
Right.
One paper caught my eye because it focuses on a practical application, while the other uses a captivating case study to reveal insights into extreme longevity.
Okay, I like that. Let's hear it.
Okay, so the first one, engineered extracellular vesicles with sequential cell recruitment and osteogenic functions to effectively promote senescent bone repair. Quite a mouthful. And it was published in the Journal of Nanobiotechnology on February 13th.
Okay.
It's by Lei Ji and colleagues from Shanghai 9th People's Hospital.
Okay.
And they're using engineered extracellular vesicles, those tiny packages released by cells that can carry genetic material and proteins to repair aging bone.
Wow, that's amazing.
I know. And what makes this paper stand out is its focus on a specific therapeutic goal: bone repair, a common issue associated with aging.
Right.
It's exciting to see researchers leveraging our understanding of epigenetic mechanisms to develop targeted therapies for age-related conditions.
Absolutely. And then the second paper, tell me about that one.
Okay, so this is a captivating bioRxiv paper from February 9th titled Epigenomic Insights into Extreme Longevity in the World's Oldest Terrestrial Animal, Jonathan.
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 Okay.
So Jonathan, a 192-year-old Aldabra giant tortoise,
A tortoise.
offers a unique window into the secrets of extreme longevity.
192 years old. That's mind-boggling.
I know.
What did the researchers learn by studying his DNA?
So the researchers from Kello Labs, they found some gene variants associated with longevity in pathways related to DNA repair and telomere regulation.
Okay. Not entirely surprising?
Right, not a huge surprise.
But what else?
But what's fascinating is they proposed a model linking efficient mitochondrial energy production to low methylation entropy in the nucleus.
Revelation entropy.
Yeah, what is that?
Yeah, and why is that significant for longevity?
Yeah, I know, right?
Break it down for us.
Okay, so it's essentially a measure of the variation in epigenetic modifications across the genome.
Okay.
Low entropy suggests a more stable and controlled epigenetic landscape.
Okay.
They believe that efficient energy production by mitochondria somehow contributes to maintaining this epigenetic stability,
Right.
which is crucial for extending lifespan. So, I mean,
That's a bold concept, connecting mitochondrial efficiency to epigenetic stability.
I know.
Could this be the key to Jonathan's remarkable longevity? And if so, are there any hints of similar mechanisms in humans? This line of research could be groundbreaking for developing interventions targeting both mitochondrial and epigenetic health.
Absolutely. I mean, I think it definitely warrants further investigation. There's a lot we can learn from these long-lived species
For sure.
that could inform our understanding of human aging.
Okay.
So for our next paper, let's shift gears and talk about a proposal that could revolutionize how we assess the effectiveness of anti-aging interventions.
Okay. Sounds good.
In vivo medical imaging for assessing geroprotective interventions in humans was published in Geroscience on February 6th.
Okay.
This paper, by Jonas E. Svensson and colleagues from the Karolinska Institutet in Sweden, advocates for a battery of medical imaging techniques to track age-related changes in various physiological systems.
Okay.
So they're talking about using MRI, PT, CT, and optical coherence tomography to monitor everything from cardiovascular health to bone density to cognitive function.
Wow.
I know.
So instead of relying solely on traditional biomarkers like blood tests,
Right.
we could have a real-time, high-resolution view of how the body is aging at the organ and tissue level.
I know. Isn't that amazing?
That's incredible. What kind of impact could this approach have on longevity research?
Oh, I think it could be transformative.
Yeah.
This approach allows for early detection of age-related pathologies,
Right.
potentially long before they manifest as diseases.
Right.
This early detection opens up the possibility of intervening much earlier in the aging process, increasing the chances of success.
That's a huge advantage.
I know.
What's more, this personalized approach could lead to more efficient clinical trials for geroprotective compounds.
Right.
We could see which interventions are most effective for specific individuals based on their unique aging profiles, paving the way for personalized anti-aging medicine.
Yeah.
It's like having a real-time dashboard to track aging and the effectiveness of potential therapies.
I know. Right. It's amazing.
Okay, so for the next paper, let's dive into the world of heart health, which is obviously a major concern as we age.
Yes.
A preprint from the University of Munster titled Altered Dynamics of ATP Synthase Cause Functional Irregularities in Senescent Hips Cardiomyocytes was posted on bioRxiv on February 11th.
Okay.
This paper uses hip cells, human-induced pluripotent stem cells, to investigate the molecular mechanisms behind age-related decline in heart function.
Got it.
The researchers focused on ATP synthase, a crucial enzyme found in our mitochondria that's responsible for producing ATP, the energy currency of our cells.
So they're looking at how aging impacts the very machinery that powers our heart cells.
You got it.
Okay.
And what they discovered is that in senescent cardiomyocytes,
Okay.
those are aging heart cells,
Okay.
the activity of ATP synthase decreases.
Uh-huh.
And even more intriguing,
Okay.
they found that this decrease in activity is linked to the physical organization of the enzyme within the mitochondria.
So it's not just about the enzyme itself,
Right.
but how it's structured and where it's located within those mitochondria.
That's exactly right. They used advanced imaging techniques to visualize the movement of individual ATP synthase molecules
Okay.
and discovered that in senescent cardiomyocytes, the enzyme is less mobile. They believe that this restricted movement is caused by structural changes in the mitochondria themselves, specifically in the folds of the inner membrane called cristae.
That's fascinating.
So they're linking the very architecture of our mitochondria to the decline in heart function as we age.
I know. What are the implications of that?
Well, the heart is a very energy-demanding organ.
Right.
When ATP synthase isn't functional optimally,
Yeah.
it leads to energy depletion in the cardiomyocytes, disrupting the heart's rhythm and eventually contributing to age-related heart failure.
Right.
This research suggests that interventions targeting mitochondrial health and perhaps even the organization of ATP synthase
Oh, wow.
could have a significant impact on preventing age-related heart disease.
That's an exciting prospect.
Yeah,
It seems we're starting to unravel the intricate connections between cellular structure, energy production, and organ function in the context of aging.
we're getting there.
Okay, let's move on to another paper that delves into the molecular mechanisms of aging. This one focuses on TOP3,
Okay,
or topoisomerase 3,
okay.
and it was published on February 11th in the journal Messenger RNA.
Interesting.
The researchers from the Institute of Cell Biology in Bern, Switzerland, used fruit flies,
Okay.
Drosophila melanogaster,
Uh-huh.
to investigate how this enzyme impacts aging. Now, topoisomerases are enzymes that help manage the structure of DNA, kind of like untangling and relieving stress in those long coiled strands. But TOP3 has a special trick up its sleeve. It can also work on RNA, specifically messenger RNA or mRNA,
Okay.
which carries genetic instructions from DNA to the ribosomes where proteins are made. So it's a multitasking enzyme.
A multitasking enzyme. I like it. What did they discover?
So they found that TOP3 plays a crucial role in the localization and stability of certain mRNAs within cells, specifically those that code for large, complex proteins involved in maintaining cell structure and function,
Okay.
by influencing where these mRNAs end up and how long they last.
Right.
TOP3GK essentially supports the production of vital proteins that keep cells healthy and functioning properly.
So it's like a cellular traffic controller, making sure the right instructions get to the right place at the right time.
Exactly.
Clever.
And when they disrupted TOP3GK function in the flies,
Uh-oh.
they observed premature aging, particularly in the neuromuscular junction, the connection between nerves and muscles. So this suggests that TOP3GK is important for maintaining the health of this critical connection,
Okay.
which deteriorates with age.
So this research suggests that TOP3J could be a potential target for interventions aimed at preserving neuromuscular function and slowing down age-related decline.
Yeah, exactly. It's definitely an avenue worth exploring.
For sure.
And, this research also highlights the interconnectedness of different cellular processes, from DNA and RNA to protein synthesis,
Right.
and ultimately to the health and function of tissues and organs.
It's all connected. Okay, what's next?
Okay, so speaking of tissues and organs, let's switch gears again and talk about a paper focusing on fibroblasts.
Okay.
This preprint from Eth Zurich and the Paul Scherrer Institute, titled Identifying Regulators of Aged Fibroblast Activation in 3D Tissue Models, caught my eye.
Okay.
It was published on bioRxiv on February 11th and delves into the challenge of reactivating aged fibroblasts.
Okay.
Just a quick reminder for our listeners, fibroblasts are cells found in connective tissues throughout the body.
Right?
They're essential for wound healing and producing the extracellular matrix, the scaffolding that supports cells and tissues.
Uh-huh.
As we age, fibroblasts tend to become less active and efficient.
Okay, so what are these researchers proposing?
So they've developed a 3D tissue model that allows them to study aged fibroblasts in a more realistic environment,
Okay.
which is a significant advancement in itself.
Yeah.
And using this model, they tested various pharmacological inhibitors to see which ones could reactivate these aging cells.
Okay.
They analyzed gene expression patterns and cell migration,
Right.
ultimately identifying 3 potential master regulators of fibroblast activation: FOXO1, STAT3, and PDK1.
And those are proteins involved in cell growth,
Yes.
metabolism, and stress response. Sounds like manipulating these proteins could potentially rejuvenate aged fibroblasts,
Yes.
leading to improved tissue repair and regeneration as we age.
Exactly.
That's exciting.
I know. It's amazing to think that we might be able to reverse the age-related decline in our cells, potentially restoring their youthful function.
It's a beautiful thought.
Okay, let's move on to our next paper, which takes us back to the lungs.
Back to the lungs, okay?
This one, Clotho regulates club cell senescence and differentiation in chronic obstructive pulmonary disease,
Okay.
was published in Cell Proliferation on February 11th.
Okay.
It examines the role of Clotho, the longevity protein,
Okay.
in aging lung tissue, specifically in a type of lung cell called club cells.
Club cells. Can you tell me a bit more about them? What are they? Why are they important?
Yeah, so they're found in the airways of the lungs,
Okay.
and they play a crucial role in protecting the lung tissue from damage and inflammation.
Okay.
They also produce a protein called CCSP,
Okay.
club cell secretory protein,
Okay.
which has anti-inflammatory properties.
Got it.
Think of them as the guardians of the lungs.
Okay, the guardians of the lungs. I like that. What happens to them as we age?
Unfortunately, as we age, club cells become less efficient.
Okay.
And this decline in function contributes to age-related lung diseases like COPD.
Right.
The researchers from Nanjing Medical University in China found that clotho levels decrease in aging lung tissue.
Okay.
And this decrease is associated with increased senescence,
Right.
cellular aging, and club cells.
So lower clotho means less protection for the lungs and more inflammation.
Right.
But I'm guessing the researchers dug deeper than that and uncovered a specific mechanism by which clotho influences club cell senescence.
Yes, they did.
Okay.
They found that clotho regulates the activity of another protein called HNRNPA2B1,
Okay.
which is involved in RNA processing.
Okay.
This HNRNPA2B1 protein turns out to be a key player in protecting club cells from senescence.
So it's a chain reaction.
Yes.
Clotho levels decline, HNRMPA2B1 activity decreases, leaving the lungs vulnerable to age-related diseases. That's quite a cascade of events.
Yeah.
What does this mean for potential interventions?
Well, it suggests that targeting this pathway, perhaps by boosting HNRNPA2B1 activity,
Okay.
could be a promising strategy for preventing or treating age-related lung diseases.
That makes sense.
It's exciting to see researchers unraveling the intricate mechanisms by which clotho exerts its protective effects on various tissues, including the lungs.
It really highlights how crucial clotho is for maintaining lung health as we age and suggests potential avenues for therapeutic interventions.
Yes, absolutely.
It's incredible to see how this protein, named after the Greek goddess who spins the thread of life, is really living up to its namesake.
I know, right? It's kind of poetic.
It really is. Okay, let's move on. What else do we have?
Okay, so let's move on to another paper that focuses on a critical aspect of aging, the decline in oocyte quality, which is a major factor in age-related infertility.
Makes sense.
This preprint titled Platt Safeguards Maternally Aged Oocytes Against Programmed Cell Death from Activating the ERK12 Pathway was posted on bioRxiv on February 11th.
Okay.
The researchers from the first affiliated hospital of Nanjing Medical University in China
Okay.
focused on a gene called Platt,
Okay.
which codes for an enzyme called tissue-type plasminogen activator, or TPA.
Got it.
They discovered that Platt expression decreases in oocytes as females age,
Okay.
making them more vulnerable to apoptosis, or programmed cell death, and ferroptosis, another type of cell death linked to iron accumulation.
So lower Platt levels mean a higher risk of cell death for these aging oocytes?
Right.
Okay, and that explains why fertility declines with age.
Right.
But I'm guessing they didn't stop there.
They did not stop there.
They found a way to protect those oocytes, right?
They did. They discovered that TPA activates a signaling pathway called the ERK12 pathway, which promotes cell survival and proliferation.
Okay.
When Platt expression is high, the ERK12 pathway is active, and the oocytes are protected from cell death.
Okay.
However, as females age, Platt expression declines, the ERK12 pathway loses its protective power, and the oocytes are more likely to die off.
It's like a delicate dance between Platt, TPA, and the ERK12 pathway, with age disrupting the harmony.
Yeah, exactly.
But what I find exciting is that they found that supplementing TPA in vitro during oocyte maturation could reduce the defect rate in aged oocytes and improve their quality.
I know.
They were able to rescue those aging oocytes by giving them a boost of TPA.
Yeah.
This suggests that TPA supplementation could be a potential therapeutic strategy for improving fertility in older females. Of course, more research is needed to translate these findings into safe and effective treatments for humans, but it's a promising step towards addressing age-related infertility.
It is.
It's another example of how understanding the molecular mechanisms of aging can lead to unexpected discoveries and potential interventions for age-related decline.
Right. Okay, let's delve into another preprint that explores the world of epigenetics and aging.
Okay, sounds good.
This one focuses on 5-hydroxymethylcytosine, or 5-HC,
Okay.
a chemical modification to DNA that's thought to play a role in gene regulation and potentially even in the aging process.
Interesting.
The paper, Scalable Screening of Ternary Code DNA Methylation Dynamics Associated with Human Traits, was posted on bioRxiv on February 13th.
Okay.
The researchers from the Children's Hospital of Philadelphia developed a new tool called the Methylation Screening Array, or MSA, that allows them to measure 5-HC levels across the genome in a high-throughput and cost-effective way. And what's really cool about this technology
Yeah.
is that it can distinguish between 5-ANC and another modification called 5-methylcytosine, or 5-MMC,
Okay.
which is more commonly studied.
So it's like having a more refined lens for examining the epigenetic landscape, allowing us to see the nuances of these different modifications.
Exactly.
Clever. What did they learn about 5-AMNC and its relationship to aging?
So they found that 5-AMNC levels change with age in different tissues,
Okay.
and these changes are distinct from those observed for 5-AMC,
Okay.
meaning 5-AMC has its own unique aging signature independent of 5-AMC.
So it's not just a passenger.
Right.
It might be an active driver shaping the epigenetic landscape as we age.
Right.
That's fascinating.
I know. And this new technology is opening up exciting possibilities for exploring the role of 5-AMC in aging and age-related diseases.
Right.
It's a step towards a more nuanced understanding of the epigenome and its contribution to longevity.
That's really cool.
I know, right? It's amazing how technology is allowing us to probe the complexities of aging at an ever finer resolution.
For sure.
It seems like we're on the verge of uncovering some truly groundbreaking discoveries in the coming years.
Absolutely. I can't wait to see what comes next. What else have you got?
Speaking of which, let's move on to a paper that explores the concept of biological age,
Okay.
which is central to the field of longevity research.
Right.
Chronological age, the number of years we've lived, doesn't always reflect our true biological age.
Right.
Some people age faster than others,
Yeah.
while some seem to defy time and maintain their youthful vigor for longer.
Of course.
And researchers are constantly seeking ways to measure biological age more accurately.
Right.
And this paper offers an interesting approach.
Okay, what is it?
The paper, Uncertainty Quantification in Epigenetic Clocks via Conformalized Quantile Regression,
Okay.
was published on Medrix Eve on February 10th.
Got it.
The researchers from Rutgers University
Okay.
are focusing on epigenetic clocks,
Uh-huh.
which use patterns of DNA refilation to estimate an individual's biological age.
Right.
They've shown promise in predicting various health outcomes and even mortality,
Sure.
but as with any prediction model, there's always a level of uncertainty.
Of course. And that's where this paper comes in.
Right.
They've developed a new statistical approach that allows them to quantify the uncertainty associated with epigenetic clocks more accurately.
Exactly.
Instead of just getting a single number for biological age,
Right.
we can now see a range of possible values, along with a measure of how confident we are in that range.
So it's about increasing the precision and reliability of our measurements,
Right.
so that we can develop more effective strategies for promoting longevity and healthy aging.
This is a big step forward for the field of biological age research.
I agree. It's all about refining our tools and approaches to gain a deeper understanding of the aging process and develop more targeted interventions.
I like it. Keep going.
Okay, let's turn our attention to a paper that explores the impact of exercise on aging hearts.
Okay, good one.
You know, we know that exercise is beneficial for overall health,
Sure.
but this paper delves into the specific effects of different types of exercise on the aging heart.
Okay.
The paper, Comparison between the effect of mid-late life high-intensity interval training and continuous moderate-intensity training in old mouse hearts,
Okay.
was published in the Journals of Gerontology, Series A, on February 10th. The researchers from Sujian Medical University in China compared two types of exercise high-intensity interval training,
Okay.
or HIIT,
Right.
and continuous moderate-intensity training,
Okay.
or CMIT, in aging mice.
So is one type of exercise more beneficial for aging hearts than the other?
Oh, they found that both types of exercise had beneficial effects on the aging heart.
Okay.
However, HIIT seemed to offer a slight edge when it came to improving cardiac function and survival rates.
Interesting.
Those short bursts of intense exercise, followed by periods of recovery,
Yeah.
might be a more effective strategy for maintaining heart health as we age.
It's encouraging to see that even starting exercise in mid to late life can have a positive impact on heart health.
I know, right?
It's never too late to reap the benefits of exercise.
Right.
And this study provides further evidence that exercise can be a powerful tool for promoting healthy aging.
Exactly. Okay, let's switch gears once again and explore a paper that delves into the complex relationship between reproductive history and aging in women.
Okay, this is an area of research that's gaining more attention as we recognize the diverse factors that influence how we age.
Right.
I'm curious, tell me more.
The paper, Evaluation of Reproductive Profiles, Epigenetic Aging, and Mortality in Postmenopausal Women, was published on Medrix Eve on February 8th.
Okay.
The researchers from Duke University used data from a large national survey to investigate the connection between reproductive profiles,
Okay.
things like age at first period, number of pregnancies, and age at menopause,
Okay.
and both epigenetic aging and mortality in postmenopausal women.
Did they find any link between certain reproductive profiles and faster aging or an increased risk of mortality?
They did. They found that women who experienced early menopause,
Okay.
defined as before age 45,
Right.
had an accelerated pace of aging as measured by a specific epigenetic clock called Dunedin-Poam.
Dunedin-Poam.
Yeah. And early menopause is concerning because faster aging is associated with a higher risk of age-related diseases and mortality.
So early menopause is a red flag for potential health issues later in life.
Right.
Did they find any connection between hormone replacement therapy and aging?
They did.
Okay.
And it was a bit surprising.
Oh.
They found that the association between early menopause and accelerated aging was particularly strong among women who had used hormone replacement therapy.
That's unexpected.
Right.
We often think of hormone replacement therapy as a way to mitigate the negative effects of menopause.
Right, exactly.
That's interesting.
It is. And, it highlights the complexity of this area of research.
Right.
It's possible that the type or timing of hormone therapy might influence epigenetic aging trajectories.
Okay.
More research is definitely needed to understand this relationship fully.
It seems there's more to the story than meets the eye when it comes to managing menopause and its long-term health implications.
Right. This study underscores the importance of considering reproductive history when assessing an individual's risk of age-related diseases and mortality. It also suggests that personalized approaches to managing menopause,
Okay.
including hormone therapy,
Right.
might be necessary to optimize healthy aging.
It's about recognizing that aging is a highly individualized process influenced by a multitude of factors throughout our lives.
Right. There's no one-size-fits-all approach to healthy aging.
Exactly.
We need to consider the individual's unique history and circumstances to develop tailored strategies for promoting longevity and well-being.
Well said. Okay, let's get back to the gut microbiome.
Okay, yes, let's return to the fascinating world of the gut microbiome.
With a paper that offers a comprehensive view of its changes throughout the lifespan.
Yes, exactly.
Okay, let's hear it.
The paper, High-Resolution Temporal Profiling of the Gut Microbiome Through the Whole Life of Mice, was published on bioRxiv on February 6th.
Okay.
The researchers, from Riken, conducted a meticulous study tracking changes in the gut microbiome of mice throughout their entire lifespan.
Okay.
Over 1,000 days of data.
Wow, that's an impressive amount of data.
I know.
They must have uncovered some fascinating insights into the dynamics of the gut microbiome over time.
They did.
Okay.
They found that the gut microbiome is incredibly dynamic throughout the lifespan.
Okay.
It doesn't just change in composition, the types of bacteria present,
Right.
but also in its stability.
Okay.
They observed that some bacterial species are incredibly persistent, present throughout the lifespan, while others are more transient, coming and going over time.
So there's a core microbiome that seems to be essential for life, while other species come and go depending on various factors like diet, environment, or even age.
Yeah,
Did they identify any particular species that were consistently present?
they did.
Oh, wow.
They even identified a core set of bacterial species,
Okay.
the life core species, which were found in at least 80% of the mice across their entire lifespan.
Okay.
This suggests that these species might play a fundamental role in maintaining health and longevity.
That makes sense. What about the connection between microbiome diversity and lifespan? Did they find any correlation there?
They did,
Okay.
and it's quite intriguing.
Okay.
In middle-aged mice, greater diversity was associated with longer lifespans.
Okay.
However, this relationship flipped in older mice, where greater diversity was actually linked to shorter lifespans.
Wait, so more diversity isn't always a good thing. That's counterintuitive.
I know, right?
It really challenges our assumptions about the role of the gut microbiome in aging.
Right?
We often think about promoting diversity as a way to maintain a healthy gut.
Yeah.
But this study suggests that the relationship is far more complex.
Yeah.
And that it changes throughout the lifespan.
Yeah. That's definitely food for thought.
It is. It seems like the gut microbiome is a key piece of the aging puzzle, but we still have much to learn about its intricate dance with longevity.
I agree. The gut microbiome is a complex and dynamic ecosystem,
Right.
and its influence on aging is multifaceted.
Sure.
This research highlights the need for a more nuanced understanding of how the microbiome evolves throughout life and how these changes impact health and longevity.
Absolutely. What's next?
Let's turn our attention to a paper that explores the potential of artificial intelligence in longevity research.
Okay, AI is revolutionizing so many fields, and it's exciting to see its application in unraveling the complexities of aging.
Yes, exactly.
What have you got?
The paper, Interpretable Deep Learning of Single-Cell and Epigenetic Data, reveals novel molecular insights in aging,
Okay.
was published in Deep Learning on February 12th.
Got it.
The researchers from the Minbo Institute of Digital Twin in China developed a novel deep learning framework to analyze large data sets of single-cell and epigenetic data. And what's unique about this framework is that it's not just a black box that spits out results.
Right.
It's designed to be interpretable,
Okay.
meaning we can understand how the AI is making its predictions.
So it's not just about finding patterns, but about understanding the biological mechanisms behind those patterns. That's incredibly valuable.
Yes, exactly. What kind of insights did they uncover using this approach?
Okay, lay it on me.
So by analyzing a massive data set of over 1 million immune cells from nearly 1,000 individuals,
Okay.
they identified a subnetwork of genes involved in ribosome function that seems to be closely linked to aging. They also applied the same deep learning framework to DNA methylation data from monocytes,
Okay.
a type of immune cell. and uncovered a pathway involved in the inflammatory response that becomes dysregulated with age.
So they're finding connections between specific molecular pathways and the aging process.
Right.
And this knowledge could lead to new targets for interventions aimed at slowing down or reversing age-related decline.
Exactly.
This paper really highlights the power of AI to accelerate our understanding of aging and to uncover novel therapeutic targets.
Right.
It's a glimpse into the future of longevity research, where AI will play an increasingly prominent role.
Oh, I agree completely. It's exciting to see how these different disciplines are converging to push the boundaries of longevity research.
Absolutely. What's next?
Speaking of pushing boundaries, let's dive into a paper that explores a rather radical approach to extending lifespan,
Okay.
reducing the activity of an enzyme called DNA topoisomerase top 2.
This is a bold idea that challenges conventional thinking about aging. Tell me more.
The paper, Reduction of DNA topoisomerase top 2 reprograms the epigenetic landscape and extends health and lifespan across species,
Okay.
was published on February 13th in Aging Cell.
Got it.
The researchers from Sichuan Provincial People's Hospital and the University of Electronic Science and Technology of China conducted experiments in yeast, worms, and mice,
Okay.
demonstrating that reducing top 2 activity could actually extend lifespan and health span across these diverse species.
That's a remarkable finding. How does reducing top 2 activity actually slow down aging?
So they found that top 2 reduction leads to changes in the epigenetic landscape,
Okay.
essentially rewiring the patterns of gene expression.
Okay.
These changes seem to mitigate some of the hallmarks of aging, including cellular senescence,
Right.
DNA damage, and dysregulated nutrient sensing.
So by tweaking the activity of this one enzyme, they were able to influence multiple processes associated with aging? That's impressive. But hold on, isn't top 2 an essential enzyme involved in DNA replication and transcription?
Yes,
Wouldn't reducing its activity have negative consequences?
that's a valid concern.
Right.
And it's something the researchers addressed in their study.
Okay.
They found that while completely eliminating top 2 is detrimental,
Right.
partially reducing its activity seemed to have a net positive effect on lifespan and health span.
Okay.
It's a delicate balance, but their research suggests that there's a sweet spot where top 2 reduction can be beneficial.
It's a bold approach that requires careful consideration, but the potential rewards are significant.
Yeah,
If we can find safe and effective ways to modulate top 2 activity, it could open up entirely new avenues for extending lifespan and health span.
it's definitely a promising area for future research.
I agree. Okay, so shifting gears a little bit, what about this paper on the aging ovary?
Yes. The ovary is a fascinating organ that undergoes significant changes with age.
Right?
And this age-related decline in ovarian function is a major driver of infertility in women.
Of course.
So what new insights does this paper offer into the aging ovary?
I'm ready, tell me.
This paper, a multi-omic single-cell landscape of the aging mouse ovary,
Okay,
was published in Geroscience on February 13th.
got it.
The researchers from Zhejiang University School of Medicine in China used a combination of single-cell RNA sequencing and single-cell ATAC sequencing to create a detailed map of the molecular changes that occur in the ovary as it ages.
Just a quick reminder for our listeners, these sequencing techniques allow us to analyze the gene expression and chromatin accessibility, respectively, of individual cells,
Yes.
providing an incredibly detailed view of cellular heterogeneity and function.
Right.
So they were able to see how different cell types within the ovary age differently and identify the specific genes and regulatory elements involved in these changes.
Exactly.
That's incredible. What did they find?
They identified several key molecular pathways that are altered with age in different cell types within the ovary.
Okay.
For instance, they found that the TGF-beta signaling pathway is dysregulated in mesenchymal cells, which are involved in supporting the structure and function of the ovary.
Right, and TGF-beta is involved in a wide range of cellular processes, including cell growth,
Yes.
differentiation, and even apoptosis.
Yes.
So dysregulation of this pathway could have cascading effects on ovarian function.
Exactly.
What else did they find?
They also found that endoplasmic reticulum stress increases in granulosa cells,
Okay.
which are crucial for oocyte development.
Right, the endoplasmic reticulum is involved in protein folding and transport,
Yes.
and when it gets stressed, it can trigger cell death.
Right.
So this finding suggests that age-related stress in granulosa cells could contribute to the decline in oocyte quality.
Yes. What are the implications of this research?
Okay.
Well, what's particularly valuable about this study is that they identified specific genes and regulatory elements that drive these age-related changes.
Okay.
This knowledge could pave the way for targeted therapies to preserve ovarian function and potentially even extend reproductive lifespan.
It's incredible to think that we might be able to intervene in the aging process at the cellular level, not just to treat disease,
Right.
but to enhance function and potentially even expand the window of reproductive potential.
It's pretty mind-blowing.
It is. Okay, what's our next paper?
Okay, let's switch gears once again and explore a paper that focuses on cognitive decline,
Okay,
a major concern associated with aging.
of course, a big one.
This paper, Astrocytic Heaven Sparkle 1, regulates cognitive decline in pathological and normal brain aging, was published in Aging Cell on February 13th.
Got it.
It investigates the role of a protein called Heaven,
Okay,
also known as Sparkle 1,
okay,
in protecting against age-related cognitive decline.
okay,
The researchers from the Federal University of Rio de Janeiro in Brazil
okay.
focused on astrocytes, which are star-shaped glial cells that play a crucial role in supporting neurons in the brain.
Yeah, the support system for the neurons.
Exactly.
What did they discover about heaven and its role in cognitive aging?
They found that increasing Heaven levels in astrocytes can actually mitigate cognitive decline
Okay.
in both normal aging and in a mouse model of Alzheimer's disease.
So heaven acts like a shield for the brain, protecting it from age-related damage.
Yeah, how does it actually work?
Yeah, I'm curious about that.
They discover that the Heaven influences the communication between neurons,
Okay.
strengthening the connections between synapses, the junctions where neurons transmit signals to each other.
So it's like bolstering the infrastructure of the brain,
Right,
making those connections more robust and resistant to age-related deterioration.
exactly.
That's amazing.
And what's remarkable is that they observed these protective effects of Heaven,
Okay.
even in the presence of amyloid plaques, the hallmark of Alzheimer's disease.
So even when the brain is under attack from a neurodegenerative disease, heaven can still provide some level of protection. That's incredibly encouraging.
Right.
It is.
This suggests that targeting Heaven could be a promising therapeutic strategy for both preventing and treating age-related cognitive decline, even in the context of diseases like Alzheimer's.
That's a really promising finding.
I know, right? It's another example of how understanding the molecular mechanisms of aging can lead to potential interventions for preserving brain health and function as we age.
Totally. Okay, what's next?
Okay, ready for our next paper.
Hit me.
This one delves into the world of microvascular aging,
Okay.
the age-related changes in the tiny blood vessels that supply our tissues and organs.
This is a crucial area of research because those tiny blood vessels play a vital role in delivering oxygen and nutrients to our cells and removing waste products. As they age, their function declines, and this can contribute to a wide range of age-related diseases.
The paper, CXCL9, modulates aging-associated microvascular metabolic and angiogenic dysfunctions in subcutaneous adipose tissue, was published in Chemokine CXCL9 on February 12.
Okay.
The researchers from the Chinese Academy of Medical Sciences and Peking Union Medical College focused on a specific protein called CXCL9 and its role in microvascular aging in adipose tissue, the fatty tissue found throughout the body. They found that CXCL9 levels increase in aging adipose tissue, and this increase is associated with impaired function of endothelial cells, the cells that line the blood vessels. So higher CXCL9 levels mean trouble for those blood vessels.
They dug deeper into the mechanisms behind this dysfunction,
Okay.
finding that CXCL9 disrupts the energy metabolism of endothelial cells and impairs angiogenesis, the process of forming new blood vessels.
So it's a double whammy. CXL9 not only impairs the function of existing blood vessels, but it also hinders the formation of new ones.
Right.
That's a recipe for trouble,
Uh-huh.
especially in tissues that rely heavily on a robust blood supply, like the brain and heart.
Exactly.
It's a bit scary.
It is a little bit, but what's promising is that they propose CXL9 as a potential biomarker for microvascular aging.
Oh, okay.
We could track its levels in the blood to assess an individual's vascular health and risk of age-related diseases.
That's incredibly valuable. It could allow for earlier intervention and potentially even prevention of those diseases.
Right.
And it underscores the importance of understanding the molecular mechanisms behind aging
Absolutely.
so that we can develop targeted therapies to address these underlying processes.
I completely agree. Okay, moving on.
Okay, let's shift our focus to another vital organ susceptible to age-related decline, the kidney.
The kidneys are essential for filtering waste products from the blood and maintaining fluid balance in the body.
Right.
As we age, their function gradually declines, putting us at risk for chronic kidney disease and other health problems.
Right. This review article, Endothelial Dysfunction in the Aging Kidney, was published on February 13th in the American Journal of Physiology Renal Physiology, and it really caught my eye.
Okay.
The researchers from KU Leuven and VIB in Belgium
Okay.
provide a comprehensive overview of the structural health functional changes that occur in the kidney as it ages,
Okay.
focusing on endothelial dysfunction.
Right.
They emphasize that endothelial dysfunction is a key driver of age-related decline in kidney function.
Makes sense.
They delve into the molecular and metabolic changes that occur in endothelial cells as they age, leading to impaired blood flow, reduced filtration capacity, and increased susceptibility to injury.
So aging blood vessels lead to impaired kidney function,
Right.
which further accelerates the aging process in the kidneys. It's a vicious cycle.
Yeah.
But what's promising is that they discuss potential therapeutic strategies for rejuvenating renal endothelial cells.
Oh,
They highlight interventions that target specific molecular pathways involved in cellular senescence and metabolic dysfunction.
so instead of just treating the symptoms of kidney disease,
Right.
we could target the underlying processes of aging in the blood vessels to slow down or even reverse the decline in kidney function.
That's the goal. This review article provides a roadmap for future research and potential therapeutic interventions aimed at preserving kidney health and function as we age.
Yeah,
It's a shift from simply extending lifespan to improving health span,
right.
the number of years we live in good health and vitality.
It's exciting to see this shift in focus towards improving the quality of life as we age,
I agree.
not just the quantity.
Okay, what's next?
Speaking of cellular quality,
Right.
let's dive into a paper that explores ribophagy,
Okay.
a cellular process involved in recycling ribosomes.
Ribophagy is essential for maintaining the quality and efficiency of protein synthesis.
Right.
As ribosomes age, they can become damaged or dysfunctional, leading to errors in protein production. Ribophagy helps clear out these old, worn-out ribosomes, making way for new, functional ones.
It's like a cellular spring cleaning for those protein-making machines.
Exactly. This paper, RPL12, is a conserved ribophagy receptor published in Nature Cell Biology on February 13th,
Okay.
identifies a key player in this process,
Okay.
a protein called RPL12.
Okay, the researchers are from Zhejiang University School of Medicine in China.
Okay.
What did they discover about RPL12 and its role in ribophagy?
So they found that RPL12 acts as a receptor for ribophagy,
Okay.
meaning it helps identify and target those old ribosomes for degradation. It's like a cellular garbage collector, tagging those ribosomes for removal.
Okay.
Without RPL12,
Right.
those old ribosomes would just hang around,
Yeah.
potentially causing problems in the cell.
They demonstrated that disrupting RPL12 function leads to the accumulation of ribosomal proteins and rRNA,
Right.
which can disrupt cellular processes and even contribute to cell death.
Right.
But how does this connect to longevity?
That's a good question. What did they find?
So they found that ribophagy deficiency accelerates cell death and impairs growth and development in both worms and fruit flies.
Okay.
Even more intriguing, they observed that ribophagy deficiency leads to age-related motor impairments in flies, while increasing RPL12 levels improve these motor defects.
So boosting ribophagy could potentially protect against age-related decline in motor function
Right.
and perhaps even extend lifespan.
It's a promising area of research.
For sure.
This paper highlights the importance of cellular recycling processes in maintaining health and longevity.
Right.
Interventions targeting ribophagy could be a novel approach to slowing down the aging process and preventing age-related diseases.
Absolutely. I like it. What else do we have?
Okay, let's move on to a topic that's often overlooked in longevity research: sleep.
Sleep is essential for restoring and rejuvenating our bodies and minds.
Right.
Disruptions in sleep patterns are linked to a wide range of health problems, including age-related diseases.
Right? This paper, a multimodal sleep foundation model developed with 500K hours of sleep recordings for disease predictions, published on mid-ARC February 6, really caught my eye. 500,000 hours of sleep data, that's incredible.
That is a massive data set.
The researchers from Stanford University used this wealth of information to develop a sophisticated AI model called SleepFM.
SleepFM.
Yeah, which can analyze polysomnography data and predict future disease risk.
So they're using AI to decode the secrets hidden within our sleep patterns.
Yes, exactly. What kind of diseases can they predict using this model?
I'm curious, tell me.
They found that SleepFM could accurately predict the risk of over 130 diseases.
Wow, over 130, that's amazing.
I know, right?
What kind of diseases?
Including heart failure, chronic kidney disease, dementia, stroke, and even death.
Wow, that's astonishing. Our sleep patterns hold valuable clues about our future health.
Right.
And it seems SleepFM is remarkably accurate in deciphering those clues.
Yeah.
How does its predictive ability compare to traditional risk factors?
Well, it outperformed traditional risk factors like age, sex, and body mass index in predicting some diseases.
Interesting. So it's not just how long we sleep,
Right.
it's the quality and patterns within our sleep that matter.
Yes, exactly.
This research highlights the importance of prioritizing sleep for maintaining health and longevity.
Right.
It's a reminder that we shouldn't underestimate the power of a good night's rest.
It's a wake-up call for all of us to pay more attention to our sleep habits.
It is, for sure. What about our final paper?
Okay, for our final paper today, let's return to the realm of DNA methylation and epigenetic clocks.
Okay, I like it.
This paper, a superlearner-based pipeline for the development of DNA methylation-derived predictors of phenotypic traits, published in DNA Methylation on February 6th, introduces a novel approach to developing more accurate and robust epigenetic clocks.
Okay, tell me more.
The researchers from Stanford University School of Medicine created a pipeline that uses a machine learning technique called superlearner to combine multiple prediction models.
Yep.
Traditional epigenetic clocks often rely on a single statistical model, which can limit their accuracy and generalizability.
Right.
This new pipeline leverages the power of multiple models to create a more robust and reliable predictor.
It's like having a team of experts, each with their own perspective, working together to create a more accurate picture of biological age.
Exactly. How well does this approach work in practice?
Good question. Fill me in.
Well, their approach has shown promising results in predicting a range of phenotypic traits, including age, smoking status, and even body mass index.
Okay.
It suggests that this superlearner-based pipeline could be a valuable tool for developing more accurate and personalized epigenetic clocks,
Right.
leading to better predictions of health outcomes and more targeted interventions for promoting longevity.
This is a significant step towards personalized medicine and could revolutionize how we approach preventative healthcare.
Okay, let's take a moment to catch our breath and reflect on the incredible research I've covered today.
Okay.
We'll be back in a moment to wrap things up.
It's truly remarkable how much progress we're seeing in the field of longevity research. We're seeing an explosion of new discoveries driven by advances in technology, a deeper understanding of the biology of aging, and a growing recognition of the importance of health span. It's an exciting time to be involved in this field, with so much potential for breakthroughs that could transform how we age and extend our health span.
I agree. It's inspiring to see the dedication and ingenuity of researchers around the world who are working tirelessly to unravel the mysteries of aging and develop interventions that could allow us to live longer, healthier, and more fulfilling lives.
And it's important to remember that this research isn't just about extending lifespan. It's about improving the quality of those years. It's about empowering people to age gracefully, maintain their independence, and enjoy their later years to the fullest.
Well said. Thank you for joining me on this deep dive into the latest longevity research. We'll continue to explore the cutting edge of this fascinating field in future episodes, bringing you the most exciting and impactful discoveries that could shape the future of aging.
Until next time, keep exploring, keep questioning, and keep pushing the boundaries of what's possible in the quest for longevity. 3280,